Now let's take a look at sample problem 2 for blood type. In this problem, unlike problem 1, we're adding in a second trait. We have the blood type trait, but we also have the trait for being Rh negative, okay, or Rh positive. Rh positive is the dominant phenotype. Rh negative is the recessive phenotype. So we have our key here. Let's consider the information that we're given in this problem. In this problem, I want to know what the expected offspring are, that gives us the clue that we're doing the cross, of a woman who's Rh negative and a man who's Rh positive. The man is heterozygous. So let's take a look at the woman who is Rh negative. Since Rh negative is a recessive trait, in order to be Rh negative, we need 2 copies of the recessive allele. So little r, little r. The man's genotype is Rh positive, and we're also told that it's heterozygous, which means we need 1 copy of each allele. Since Rh positive is the dominant phenotype, having even 1 allele, the Rh positive allele, gives us an Rh positive phenotype. Principle of segregation, the alleles will separate during meiosis. If the woman's 2 alleles separate into eggs, we would have a little r egg and a little r egg. If the man's gametes separate, we would have a capital R and a lowercase r. So let's take a second now and let's draw our Punnett square and look at the children. All right, I have my Punnett square drawn, so I'm going to put one parent's gametes across the top. We'll put mom's here, and we'll put the man's gametes down the side. Next, I need to go in and fill out my box, do my combination for fertilization. And I can see the results of my cross then. Both of these top boxes are the same. They're heterozygous for Rh negative, Rh positive. That tells me that because I have at least 1 copy of that dominant allele, that these 2 individuals are both Rh positive. The bottom 2 boxes are both homozygous recessive. Homozygous recessive is always Rh negative. Therefore, 50% of my offspring are Rh positive, and 50% of my offspring are Rh negative.